Assalamu alaikum. My name is Professor Triwibowo Yuwono from Gadjah Mada University, Yogyakarta, Indonesia. Welcome to our presentation on genomic organization, where we will explore the fascinating architecture, complexity, and evolution of genomes. Our journey will take us from the simplest prokaryotic life forms to the intricate structures found in human DNA, providing the comparative analysis of how genetic material is organized and how it has evolved over billions of years. Let's begin by distinguishing between the two fundamental types of cellular life, prokaryotes and eukaryotes. In the prokaryotic territory, such as bacteria, the genome typically consists of a single main circular chromosome located in a region called the nucleoid, often supplemented by smaller, circular DNA molecules known as plasmids. In contrast, the eukaryotic territory, encompassing organisms from fungi to humans, features a more complex genomic arrangement. Here, the primary genome is housed within a membrane-bound nucleus, organized into multiple linear chromosomes. Additionally, eukaryotes possess extrachromosomal DNA in organelles like mitochondria, and in plants, chloroplasts. The prokaryotic genome, despite its relative simplicity, is a marvel of efficiency. Its architecture is characterized by DNA arranged in a dense clump, the nucleoid, forming a closed, circular double helix. Functionally, genes are often grouped into single transcriptional units called operons, allowing for coordinated regulation. This organization contributes to remarkable efficiency, with genes arranged closely together and very little non-coding or intergenic space. Replication is equally streamlined, involving bidirectional replication followed almost immediately by binary fission, enabling rapid reproduction. To further solidify our understanding, let's look at a direct comparison. Prokaryotes typically have a single, circular chromosome, often with plasmids, composed of naked DNA. This genetic material floats freely in the cytoplasm. Their cell division is a rapid process of binary fission. Eukaryotes, on the other hand, possess many linear chromosomes, where DNA is intricately coiled around histone proteins to form chromatin. Their genetic material is contained within a nucleus, and cell division involves a more complex process of mitosis, ensuring precise organization into two equal groups. Now, let's delve into one of the intriguing mysteries of genomics, the C-value paradox. The C-value refers to the total amount of DNA in a haploid genome. Surprisingly, this value does not consistently correlate with an organism's perceived genetic complexity. For instance, a fruit fly has a genome of about 165 megabases, while humans have a much larger 3,000 megabase genome. However, a hermit crab boasts an enormous 149,000 megabase genome, and the plant Paris japonica as an even larger one. This raises a critical question. If genome size doesn't directly equate to complexity, what accounts for these vast differences? The resolution to the C-value paradox lies in understanding gene density. When we compare bacteria, archaea, and eukarya, we observe significant differences. Bacteria and archaea generally have smaller genomes, 1 to 6 megabases, with very high to high gene density, meaning most of their DNA codes for proteins and introns are largely absent. Eukarya, however, exhibit a wide range of genome sizes, from 10 to 4,000 plus megabases, and crucially, very low gene density. This lower density is attributed to what we call the dark matter of the genome, massive amounts of non-coding repetitive DNA, and intervening sequences, or introns, which are present in most multicellular eukaryotic genes. It's these non-coding regions that contribute significantly to the larger genome sizes without necessarily increasing the number of functional genes. Let's take a closer look at the anatomy of the human genome, which comprises approximately 3.2 billion base pairs. A striking insight is that a staggering 98.5% of the human genome does not code for proteins, ribosomal RNAs, or transfer RNAs. Only about 1.5% consists of exons, the blueprint that codes for proteins. The intermediaries, introns, make up about 20% along with regulatory sequences. Transposons, or jumping genes, account for a significant 44%, including elements like L1 and ALU. The remainder is composed of unique non-coding DNA, 15%, and repetitive DNA unrelated to transposable elements, 14%. This highlights the vast complexity beyond just protein-coding genes. The architectural differences between prokaryotic and eukaryotic genes are profound. Prokaryotic genes are typically non-interrupted, meaning they lack introns, and are often organized into polycistronic operons. This allows a single messenger RNA molecule to code for multiple polypeptides. In contrast, eukaryotic genes are interrupted, characterized by coding regions called exons interspersed with non-coding introns. These genes are monocistronic, meaning one mRNA codes for one polypeptide, and require extensive mRNA processing to remove the introns before translation can occur. While the iconic B-form double helix is the most common representation of DNA, it's important to recognize that DNA can adopt various structural variations. The B form, our standard, is right-handed with 10 base pairs per turn and a 19 angstrom diameter, typically found under low salt conditions. However, under high concentrations of potassium, sodium, or cesium ions, DNA can transition to the A form, which is also right-handed but more compact, with 11 base pairs per turn and a 23 angstrom diameter. Interestingly, DNA can also form a left-handed helix known as the Z form, characterized by 12 base pairs per turn and an 18 angstrom diameter. This Z form is stable in high salt and when cytosine residues are methylated, suggesting potential regulatory roles. One of the most remarkable feats of genomic organization is the packaging of an immense amount of DNA into the confines of a cell nucleus. This involves a sophisticated compaction hierarchy. The fundamental unit is the DNA double helix, which is 2 nanometers in diameter. This helix then wraps around histone proteins to form nucleosomes, creating an 11 nanometer beads on a string structure. These nucleosomes further coil into a 30 nanometer chromatin fiber. This fiber then forms loops, anchored by a protein scaffold, eventually leading to the highly condensed 1 micrometer diameter chromosome visible during cell division. This intricate packaging is essential for fitting the entire genome within the nucleus and for regulating gene expression. Eukaryotic chromosomes are not just simple strands of DNA. They possess distinct landmarks, crucial for their function and stability. Each chromosome has 2 arms, the shorter P arm, petite, and the longer Q arm. The centromere is a constricted region that separates these arms and serves as the attachment point for spindle microtubules during cell division, ensuring accurate chromosome segregation. At the very ends of the chromosomes are the telomeres, protective caps formed by repetitive nucleotide sequences, typically TTA-GGG in humans. Telomeres safeguard the genetic information during replication and prevent chromosome degradation, with their maintenance often linked to the enzyme telomerase. Evolutionary genomics reveals profound insights into how minor differences in gene sequence or regulation can lead to striking differences in physical form. A prime example is the EVO-DEVO toolkit, particularly the homeobox or HOX genes. These genes are highly conserved across diverse species and play a fundamental role in controlling the body plan and development of embryos. The remarkable conservation of these genes across evolutionary time underscores their critical importance as a universal developmental toolkit, demonstrating how small changes in their expression or regulation can drive significant evolutionary innovation. When comparing close relatives, such as humans and chimpanzees, we find an astonishing 99.5% gene conservation. Despite our obvious phenotypic differences, our genomes are remarkably similar. The genomic variance primarily stems from a mere 1.2% difference at single base pair positions, along with 2.7% due to insertions and deletions. A key functional impact of these subtle differences is exemplified by the FOXP2 gene, which regulates genes involved in vocalization. While the FOXP2 gene in Neanderthals is identical to modern humans, suggesting speech capability, distinctions in this gene between humans and chimpanzees are thought to contribute to our unique capacity for complex speech, highlighting how small genetic changes can have profound functional consequences. Even within a single species, such as modern humans, there is significant genomic variation, albeit relatively low given that humans have existed for approximately 200,000 years. This variation manifests in several forms. Single nucleotide polymorphisms, or SNPs, are single base pair changes in the DNA sequence. Structural mutations involve larger physical alterations to the chromosome, including inversions, deletions, and duplications. Finally, copy number variants, or CNVs, are large segments of the genome that are repeated a variable number of times across different individuals. These variations are highly impactful for human evolution, disease susceptibility, and individual traits. In synthesis, we can view the genome as a layered ecosystem governed by several overarching principles. Firstly, architecture over size. The C-value paradox clearly demonstrates that organismal complexity is driven not simply by raw gene count, but by gene regulation, alternative splicing, and the massive non-coding dark matter regions. Secondly, packaging constraints. The physical necessity of condensing a 2-nanometer DNA string into a 1-micrometer chromosome dictates complex cellular behavior and requires intricate structural landmarks like centromeres and telomeres. And finally, evolutionary conservation. From highly conserved Hox gene toolkits governing body plans to the FOXP2 speech gene, the genome acts as a historical record, constantly reusing and modifying ancient code to build modern complexity. Understanding these layers is key to unraveling the mysteries of life itself. I have described the very foundation of genome architecture in organisms. I hope that you enjoy my lecture. Thank you and see you again in my next lecture.